Testing, testing. Maybe I do need a laptop. This keeps slipping. Anyone got a laptop I could borrow? This does keep slipping a bit. Oh, yeah. Thanks. It's all right. All right, that's better. Okay. So, good job on exam 2. You guys did really well. The average was like, before extra credit in the curve, the average was like a 77 or something like that, which is really, which is quite a few points higher than it was the last couple semesters, so good job. So, what I'll start with today, since we've got plenty of time till exam 3, is, does anyone have any particular questions they want clarified about exam 2? Yes? There might not be a curve, but I always at least give a minimum of 2 points, no matter what. But it probably won't be a big curve, especially because this is before even the extra credit was calculated, so we'll see. Other questions? What do you guys think about this? I was walking around trying to clarify a couple things with everyone on exam 2. It seems like one of the most tricky ones was the one with, what was it? Which one of the following carbons is beta anomeric, right? Yeah. So notice it was not asking which carbon represents a beta link, just which one is beta anomeric. So let me go ahead and draw something here. Okay. What? All right, let me just go ahead and draw something. Oh, I messed that up. I think I have that backwards there. Okay. Oh, no, wait, maybe that's right. I might have these two carbons mixed up. I always get these two OHs mixed up. I always get fructose backwards. All right. So you were given something like this, and there were arrows pointing at a bunch of different places. I might not remember all the places. Right? And it was asking which carbon is beta anomeric. Now, it seems like the most common mistake people were looking at is they were looking at this spot right here. The first mistake they're making is they're looking at this spot right here, and they do notice that this is pointing down and this is pointing up. This does describe an alpha link. That is, if it was asking for what type of linkage, the answer would be an alpha link. The next thing, and people are getting closer, the next thing is people were looking right here, but they were making the mistake of noticing this was pointing down and this was pointing up and thinking that this was alpha, right? You're not supposed to compare it to the CH2OH right next to the anomeric carbon. You're supposed to compare this thing right here with that CH2OH, the one on the opposite end there. And so because they're both pointing in the same direction, this particular carbon is beta anomeric. Okay. All right, so that seems to be one of the things a lot of people were asking about. Is that all right? Makes sense? Okay. Another one, and this one I'm pretty sure a lot of people really, really overanalyze. Like, I'm sorry, I'm sorry I probably made you guys paranoid, or hazy makes you guys paranoid, I don't know. But there is like a picture of a cell membrane with, let me attempt to draw it. I apologize for maybe scaring you guys. I don't know. Something like this. And it's asking, the picture above shows what? And it gives you several options. So, without giving you the exact answers, the answer was not all the above or anything, and it isn't really... And it wasn't even counting all of the peripheral proteins or anything. The answer was, how many integral proteins do you have here? Wow. Yeah. and some of you got it, because when I was asking around, it's like, okay, you know what integral protein means and what peripheral protein means. In your picture here, what is the only ones that are passing through the entire cell membrane and things like that? I'm trying to remind you guys that integral proteins are probably one of the easiest things to identify. Peripheral proteins, as you can see, gets really complicated, and I kind of made sure that that wasn't going to be the question here, because that's up to debate of how many peripheral proteins there were. So I made it as obvious as possible. I gave you the one that probably would be easily identified. Yeah. It's asking what the picture above shows. Yeah, but I can't give you the answer choices again. Okay, well, the only correct one was 3 integral proteins. Okay. Let's see. Oh yeah, these squiggly lines each represent lipid anchor subunits. So that's plenty of them. And peripheral proteins, each one of these things is a peripheral protein. So that was another tricky one. Let's see. Oh, the Nernst equation one, that one, it seems some of you were calculating not getting any of the numbers there. When I ran into you guys, the most hint that I could give you is make sure you don't type it all in at once in your calculators, because calculators don't like it. Sometimes when you type in everything at once, you got to calculate the fraction first, then log, then multiply it afterwards, and do order of operations. Yeah. The first question? Oh, this one right here? Yeah, what about? Oh, go over it? Yeah. So this one was, which one was beta and Emeric? Not which carbon points to what type of linkage that you have. So again, the most common mistakes is people were picking this one, or were about to pick that one, because this showed, this carbon tells you what type of linkage you have. Unfortunately, this carbon is an alpha link because they are anti to each other. So that's not the correct answer. The next thing, and they were getting a little closer, is a bunch of people were picking this one. But although this is an anomeric carbon, a lot of people thought this was the wrong answer because they were comparing it to this particular spot right here. If you look at this pointing down and this pointing up, you would think that it was an alpha link, that it was a alpha anomeric, but that's not right. You're supposed to compare this thing to this thing over here, and they're both pointing down, which means it's beta anomeric. So the beta anomeric carbon is specifically this thing right here. Yes? Was it?
The picture said there are not 3 integral.
No, there are 3 integral proteins in this picture. There are 3 integral proteins in this one. That was the right answer for this one. The other statements that were given were false statements, like, I'm not going to say, but the number of lipid anchors given in the answer choices does not match what's on the picture. The number of peripheral proteins also given in the answer choices does not match the picture either. Okay. Let's see. Other ones. Oh yeah, there is one thing that I actually completely forgot to go over at the review, but it seems like based off how people responded, not many of you were tripped up by it too much. It's the long word problem talking about lactose is a disaccharide found in milk. Many people cannot digest lactose, blah, 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 blah. I guess Hasey's went over something like that in class? Okay. All right. Yes, I believe, yeah, that one was all the above. Yeah. But to go over it real quick, the basic concepts of lactose intolerance as a medical condition rather than like a genetic thing. As a medical condition, here I go, attempting to draw here. Here's your small intestine. Okay. And it's lined with intestinal cells. Okay. So if you're unable to digest lactose, lactose starts building up on the walls of intestinal cells, of the intestine. Okay? It's obviously not drawn to scale, but lactose starts building up here. We didn't go over osmosis here, but maybe you might remember in like other classes, the way osmosis works is water always flows to the side with more solutes on it because it's trying to dilute the solute. So if there's a lot of lactose sitting outside the cell and building up, that is a solute. So water from these intestinal cells are all going to flow out of the cell into the cell membrane to try to dilute all this lactose. Okay? So osmosis is causing water to flow out of the cell membrane into the small intestine to try to dilute the lactose that isn't being degraded because of this lactose intolerant person. That's one thing. Some other answer choices, well, if you're losing water, you're dehydrating both the cells and the muscles right in the layer under that as well. And the last one is a little bit more general than that, but has been talked about in Hasey's class is the fact that bacteria, if the cells aren't going to eat the bacteria, if the cells aren't going to eat the lactose, the bacteria certainly will, and a byproduct is usually methane. So all three factors contribute to the symptoms that you see for lactose intolerant people. Okay. That was the one weird looking one, weird question. The extra credits, you guys received glycogen phosphorylase as your extra credit. Hopefully y'all did good on that. I haven't graded it yet, but like I said, as long as you show me enough that you've proven to me that you know what you're talking about, you'll get the full credit. All right, let's see if there's anything else. Any other questions? Yes? Max of 5 extra credit points, yeah. Any other questions that they were wondering about the exam? Because it was only Monday, so if we wait any longer, we're going to forget what was on the exam. So, all right, we're good? Okay, so let us move on to what I consider the most important part in this class. And probably if you're going into any health field, it's the most important part. If you've taken genetics, a lot of it you may have seen before as well. So that is what is called the central dogma of molecular biology, which I'm going to go over in a second. If you've ever learned about DNA replication or transcription or translation, those are the most important concepts that you will learn in this class and the most important concepts you need to take away from this class if you're planning on taking the MCAT or any other like health-related test or field. Okay, so in fact, actually, the first thing I'm going to do is draw something. And I know we're probably moving a little bit ahead, right? Because Dr. Hasey's hasn't started lecturing this stuff yet. Okay. Well, so I may skip certain things that he goes over in lecture because I'm only going over what he is most focused on in previous semesters. He might add new stuff, so pay close attention to stuff that he adds in class. I'm only going over the stuff that I know he goes over in previous semesters. So first, most important, and man, you cannot underestimate how important it is to understand the central dogma of molecular biology. Everything that you're going to learn from this point onwards ties back into the central dogma. Use this as context to help you conceptualize certain things about molecular biology. A lot of this stuff you may have heard these terms already before. All right, so we have DNA. DNA is the basis of everything that lives, right? DNA contains all your genes that code for everything that makes who you are, makes what animals are and plants are. It all starts with DNA. But DNA by itself is not functional. DNA does not do anything itself. How biology works is that DNA is transcribed into RNA. Okay? Transcribed or transcription is one of the most important processes in molecular biology. And you basically, DNA is like a template. It is transcribed into some not yet functional form of nucleic acid, but a kind of intermediate form called RNA. You guys know about, you may have heard about messenger RNA before. It isn't the only type, but regarding this context, we'll use messenger RNA as the most important one right now. And this messenger RNA will get translated into proteins. This is the actual functional part of the central dogma of molecular biology. We already talked about enzymes, and back in exam 1, we talked about certain structural proteins like alpha keratin and beta keratin. And the enzymes we talked about, like allosterically regulated enzymes, Michaelis-Menten enzymes, are all proteins, okay? So proteins are derived from messenger RNA, which are derived from DNA. So this is how these three important areas of biology are tied together. There is one more thing, and the fact is that living systems have the capability to replicate. And that is because DNA can undergo a process called replication, okay? So all the processes we're gonna go over, the little components of each of these processes we're gonna go over, all have to do with this central dogma, starting from DNA and eventually getting all the way to proteins, which is the functional stuff of everything. For the MCAT, for this class, for any other biology course in the future, you need to conceptualize this and understand basically every component of this. So, this is chapter 28, replication. We also talk about nucleotides in chapter 10 and 11, so it's kind of spread out everywhere. But replication is chapter 28, transcription is chapter 29, and translation is chapter 30, which will be taught after exam 3. Okay, so basically everything beforehand is for exam 3, and everything after is added to the final. By the way, your final exam, 50% of your final exam will be about material that you learn from today onwards. So exam 1 and 2 put together is only 50% of the final. The remaining 50% of the final is everything that you're going to learn today until the end of the course. So from chapter 10, 28, 10, 11, 28, 29, and 30. So this is the most important stuff. All right, now we'll start with the boring stuff, and that is the nucleotides. The structural stuff, because this is a biochemistry course, not exactly a biology course, so we have to go over the structural stuff first, and then we'll go over the mechanisms. Okay, so chapter 10 is really long, really long. It's split up into 2 PowerPoints, and I probably have not included everything on here. So, you know, if there's other stuff that we run into, if you run into in lecture, I'll cover it here. But I'm only going to focus on the most important stuff, in my opinion. So, 10A is talking about central dogma, first of all, which we already talked about. Here's a different diagram of the central dogma. DNA replication, transcribed into mRNA, and mRNA is translated into proteins. So, first, the components of DNA and RNA are your nucleotides. But the nucleotides themselves are made of sugars and phosphates and these things here, which are your nitrogenous bases. So the very first thing you need to do when you're studying from this point onward is memorize these nitrogenous bases. Let me pull up a picture. Well, actually a category thing first. Your nitrogenous bases can be split up into 2 categories, pyrimidines and purines. just like with amino acids and sugars, because this is a multiple choice exam, you don't have to memorize every single double bond and every single carbon and every single nitrogen that goes everywhere. Feel free to if you want to, but you need to be able to identify the nitrogenous bases just from each other. So if you know a little bit about each one, you can figure out which one is uracil, which one is thymine or cytosine or something. So I'm going to try to give you guys some hints with that. So first type of nitrogenous base are your pyrimidines. Okay, and pyrimidines you can identify if they just have a single ring structure. Contrast that with purines, of course, that have 2 rings in them. So pyrimidines, you have 3 types: cytosine, uracil, and thymine. You can come up with stupid acronyms to help memorize these things. A long time ago, I came up with some acronym about like cut pyramid. It doesn't make any sense, but the acronym is cytosine, uracil, thymine, cut, and pyramid, pyrimidine. That's just how I remember it. Feel free to come up with whatever thing that helps you memorize these things. Someone once told me, I think it was Durbin's even, he told me he came up something with the purines. Purines are pure as gold, AG, adenosine and adenine and guanine, pure as gold, purines. But anyways, back to the structural stuff. So after you memorize which ones belong in which category, how can you tell the difference between them? You'll notice that a lot of it actually looks very similar between them. What I personally focus on, and you can use other differences, what I personally focus on is on this functional group at the top right here, or at least the 2 kind of carbons that you see up here. You should try to memorize, and this is what I do, that cytosine is a single ring group with an amine at the top there. None of these other 2 have an amine at the top. Uracil, for reasons that we're going to see later, has this carbonyl group instead. It looks very similar to cytosine, but has a carbonyl group. In fact, somewhere in the notes they'll explain why that is that spontaneously cytosine has the capability to just transform into uracil, to undergo a reaction called deamination. All right, so we'll see it again later, but cytosine, this amine group, can spontaneously deaminate, and cytosine transforms into uracil with this carbonyl group. Bottom line so far, cytosine has an amine group, uracil has a carbonyl group. Thymine should be one of the easiest things to identify because thymine, by design, looks exactly the same as uracil, but one small difference, right? If you compare thymine and uracil to each other, the one small difference is this methyl tag attached to it, okay? So, how I memorize this is cytosine has an amine group here, I don't care about everything else. Uracil has a carbonyl group at the top here, I don't care about everything else. Thymine is basically uracil, but with a methyl group attached to it, and I don't care about anything else. So, because again, this is a multiple choice exam, that's my personal shortcut to use to memorize this stuff. Feel free to come up with any shortcut that you guys prefer. Don't worry about memorizing this really complicated name regarding these nitrogenous bases. Just go with what is accepted in biology, cytosine, uracil, and thymine. For your purines, you can use similar tricks. Just look for like one difference and stick with that difference. So again, your purines are adenine and guanine. Adenine I memorized because it has an amino group here. Guanine doesn't. That's it. Guanine has a carbonyl group instead there. Adenine starts with an A. Amino group is right here at the top. Guanine instead has a carbonyl group there. Very similar to like this relationship between cytosine and uracil, where cytosine has an amino group and uracil has a carbonyl. Down here, adenine has an amino group and guanine has a carbonyl. So again, don't worry about the complicated names in parentheses, just memorize the general names and general structures. Okay. All right, so that started out pretty easy. Now we get into some slightly more complicated stuff. Good news, you don't have to memorize the structure of these things. These are what are called special RNA bases. Special RNA bases. So it turns out biology is more complicated than just being composed of 5 parts. There are many other very rare, uncommon RNA bases that look very similar to the purines and pyrimidines that we were looking at earlier. In this class, you won't have to memorize the names of these things, but if you are given any of these names, as long as you understand that they represent special or uncommon RNA bases, you're good. You don't have to memorize this picture must be hypoxanthine, this picture must be xanthine, nothing like that. As long as if you see these names, you recognize that they're special or uncommon RNA bases, that's all you need to know. What I mean by that also is that these bases are never going to be found in DNA. You will not find any of these bases in DNA at any time in development or anything. These are only going to be found in RNA. And part of that reason is that DNA has to be consistent, right? If DNA determines everything, it needs to be consistent. As consistent as possible, right? We hear about mutations occur sometimes, genetic diseases occur sometimes, whatever. But it's going to try to be as consistent as possible. DNA is going to try to remain being composed of A's, T's, G's, and C's, adenines, thymines, guanines, and cytosines. RNA, you have 4 standard bases with RNA as well, adenine, uracil, guanine, and cytosine, but of course, sometimes you have these bases show up. They serve various purposes like helping to construct ribosomes, helping to construct tRNA. You don't really see these in mRNA, but again, that might be a bit too specific information, so if you forget what I just said there, don't worry about it. We'll move right along. All right, these here are properties, known properties of your nitrogenous bases. We'll go over each one in a second. Keto-enol tautomerization, acid-base dissociation, and strong absorbance of UV light. So let's see what this means. So first, the phrase keto-enol tautomerization. In Orgo 2, you had to learn about what that means. I'll simplify it for you here. Keto-enol tautomerization is kind of like a thing where a carbonyl group can sometimes become an alcohol group and back and forth. It is way more complicated than that, but in the context of biochemistry, that's what it means, is that some of your nucleotides, this carbonyl at the top here, can sometimes transition and become an alcohol group or back into a carbonyl group depending on certain conditions in the cell. I'll even simplify it even more. This keto-enol tautomerization, this differential favorability between the ketone group and the alcohol group, it turns out that nucleotides and nitrogenous bases overwhelmingly prefer the keto form in the keto-enol tautomerization thing. That overwhelmingly, a majority of them prefer the keto form. That's what you need to know for this class. Biochem 2 and maybe the MCAT, it might not even be on the MCAT. You'll learn that it's a bit more complicated than that, that some nitrogenous bases actually prefer the enol form and only some of them prefer the keto form. But for this class, keto form is the most preferred state for keto-enol tautomerization. The keto form is the most preferred one. Moving along, oh yeah, there's, for your purines, this is what keto and the enol form look like. Same thing, keto is preferred. All right, so I guess going a little bit out of order, the second property that's important in terms of nitrogenous bases is the fact that they absorb UV light. You might remember from organic chemistry or even earlier in this class that a certain type of functional group or structure absorbs UV light. What organic chemistry 2 structure likes to absorb UV light? Aromatic rings, right? We learned your aromatic amino acids absorb UV light. Well, in the context of nucleic acids, context of nucleotides, we'll notice they all basically have aromatic rings. So it turns out that DNA and RNA, because they're made of these nitrogenous bases, will also absorb UV light. In fact, one of the most common experiments that is ran in the lab is taking your sample of unknown amount of DNA or RNA and putting it in a machine and seeing how much UV light is absorbed. Depending on certain properties, depending on how much of certain types of UV light is absorbed, your unknown solution, you can say, oh, this is either pure DNA or pure RNA. So what do I mean by that? So don't worry about memorizing this graph. Do not worry about memorizing this graph. The most important thing is this statement in red down here, and I'll explain what this means. So UV light comes in several different numbers. UV light, specifically, the most important numbers that matter in terms of UV light is at 260 nanometers, which is right here. And again, don't worry about memorizing the graph. I'm just using it to illustrate a point. So UV light, the most important parts are at 260 nanometers and 280 nanometers. The way this experiment works is they adjust the machine to 260 nanometers, and they measure how much of that light is absorbed by your unknown solution. Then they adjust the machine to 280 nanometers and remeasure how much light is absorbed. And it prints out two numbers, okay? The most important part is that they take those two numbers and they divide them by each other. Whatever the absorbance was at 260, divided by whatever the absorbance was at 280, will print out a particular number. If that number, if that ratio of absorbance equals 1.8, it means your unknown solution is pure DNA. Okay, so again, that ratio between some absorbance at 260 nanometers divided by some absorbance at 280 nanometers, it's going to calculate a ratio for you, and the machine does this automatically. And if it comes out to 1.8, it means you have pure DNA, and if it comes out to 2.0, it means you have pure RNA. Anything that is sufficiently less than 1.8, and this is some bonus knowledge, you don't really need to know this, any number that is sufficiently less than 1.8 means your unknown solution is contaminated a lot. That happens a lot in the lab. If you're trying to extract DNA or RNA, sometimes some protein is left over and it messes with all your all your experiments that you plan to do in the future. So you have to redo everything. Yes? Not to calculate it, but you may be given a statement such as the 260 to 280 ratio, if it is equal to 1.8, what do you have? The answer is DNA. If it's equal to 2.0, what do you have? The answer is RNA. And there might be some tricky things. There might be an answer choice that says the 280 to 260 ratio is 1.8. That means you have DNA. That's a false statement, right? If the answer choice says 280 to 260, that's backwards. All right, so that's why most important is to memorize this statement here, okay? So again, context. Why do we care about DNA and RNA absorbing UV light? It's one of the ways that we analyze DNA, and it's simply to, if we're given some solution, we do this experiment, we figure out the ratio, and we can figure out what we have as DNA and RNA, and we can do different experiments afterwards, okay? Let's see. Oh, if you get something that's sufficiently higher than 2.0, it means you have some type of like dust or something in solution. Again, bonus information. You don't need that for this class, but I'm helping give you context of why this can be important. So memorize this red statement. It's very important. You might see it on the exam. Okay. Before we move on, let me see. All right, let's reinforce some of this stuff, because this is supposed to be SI. So, I'm not going to ask anyone to describe the entirety of the central dogma of molecular biology, but I'm going to say some things, and you guys got to fill in the blank. So, central dogma of molecular biology starts with DNA becoming mRNA through what process? Transcription, very good. And mRNA becomes proteins through what process? Translation. The reason why life happens is because DNA can also undergo what to make more of itself? Replication. Very simple, okay? There are two types of nitrogenous bases, pyrimidines and purines. From the following structures below, identify which one it is, pyrimidine or purine, and specifically what nitrogenous base it is. And at the end, we'll do this at the end, state whether a base is found in DNA or RNA or both. So we'll start with this thing on the left. Is that a purine or a pyrimidine? It is a pyrimidine. One ring is a pyrimidine, okay? Anyone want to guess what nitrogenous base that is? Thymine, exactly. If you go with my shortcut, you look at this functional group up here, it is a carbonyl, and notice it has a nice bonus methyl group here. That makes this thing on the left thymine. Okay, making sure that's picking up. Okay. The next one here, pyrimidine or purine. Purine because it's two rings. Good. So that gives us two choices. It's either A or G. It's either adenine or guanine. Anyone want to guess what this one is? Adenine, exactly. Again, my shortcut personally is the fact that there's an amino group here. I don't memorize the rest of this stuff. Okay. All right. We know that's a purine, but what nitrogenous base must it be now? Guanine because of the carbonyl group. Let's do the one on the left here. We know that's a pyrimidine. What is it? Cytosine, and I personally memorize it because there's an amino group here, and I don't memorize anything else. And the last one, by process of elimination, must be uracil, right? Uracil is also a pyrimidine, but instead of an amino group here, it is a carbonyl group here. And notice it does not have a methyl group here, which thymine did have that, okay? All right, so we figured out which ones are purines and pyrimidines and what nitrogenous bases they are. Now we have to state whether they are found in DNA or RNA or both. So instead, I'm going to do it this way. There are 4 bases found in DNA naturally. What are the 4 bases? And you can give the letters. That's fine too. A's and T's, good. G's and C's. Adenine, thymine, guanine, and cytidine, or cytosine, are your 4 bases that you're going to find in DNA. RNA is very similar. RNA, you have A. Instead of thymine, though, you have uracil, guanine, and cytosine. So this difference actually brings up an interesting thing. And some of you might notice it, and if you don't notice it now, it'll be reinforced later, is remember when I said try to compare thymine and uracil to each other, what did you notice? That they're basically exactly the same, thymine and uracil, except for one thing, and that is this methyl group right here on thymine, right? There's a reason for that. Okay. And that's uracil over there on the right. There's a reason for that small difference there. In DNA, I said earlier that cytosine sometimes, just for random chance, spontaneously deaminates to become uracil. For no reason. Cytosine just deaminates to become uracil sometimes. That's a problem, right? Because DNA, we want it to be as consistent as possible. If it's changing all the time, it's going to lead to mutations and certain genetic diseases. So if cytosine becomes uracil, how do repair enzymes know that a mistake happened? How do repair enzymes know that even this deamination occurred in the first place? So you'll learn later when we talk about DNA replication that there's a series of enzymes that constantly go around the DNA and make sure that there aren't any mutations or mistakes in it. Sometimes it skips it still, but there are these repair enzymes are usually very good. So how does it know an error occurred here? Well, if it runs across uracil, all it's going to look for is a methyl group here, because it knows that if there was a methyl group here, it knows that what that was supposed to be was thymine. If it finds no methyl group here, it realizes, the repair enzyme realizes that this is uracil, and it understood that, oh, it used to be cytosine. So the repair enzyme will remove this thing on the right here because it lacks that methyl group marker. It's going to remove this and replace it with cytosine again in order to fix it. There are other mutations that occur that are not this, and you don't have to know about them in this class. Other mutations are taught in like genetics classes. But the one that's really important here is the fact that sometimes cytosine, just for whatever reason, becomes uracil. And the fact that uracil does not have the methyl group right here tells repair enzymes to remove it and put back cytosine. Okay? If it runs into later down the DNA strand, if it runs into something that looks like uracil, but instead it has a methyl group here, it's going to leave it alone because that methyl group tells it, all right, this is thymine, it's supposed to be here. So that's why there's a subtle difference between thymine and uracil. As for RNA, it turns out, and we'll talk about this when we get to transcription, RNA doesn't really have any repair mechanism like DNA. DNA needs to be repaired constantly because it's the most important part. RNA, if there's any issues with RNA, like if there's a mutation in RNA, it never gets fixed because RNA, you'll learn, is a temporary thing while DNA is a permanent thing. Because RNA is temporary, and if there's mistakes in RNA, what ends up happening is that you just make more RNA and maybe it'll be right this time. So that's another difference between DNA and RNA. DNA needs to be constant, so it's repaired all the time. RNA can have errors in it because if there are errors in it, they'll just remake the RNA. It'll be correct the second time. We already answered this stuff, so I'm going to skip that. True or false? Uncommon nitrogenous bases like xanthine can be found in both DNA and RNA. False, right? The uncommon ones are only found in RNA, okay? Over and over again, DNA needs to be constant. DNA has no time to be made of special nitrogenous bases, special rare nitrogenous bases. RNA, though, because it is a temporary structure, can be made of specialized uncommon nitrogenous bases. So this is a false statement because it's only found in RNA, the uncommon stuff. The difference between keto and enol forms we just saw is just basically the keto form has what functional group in it? A ketone, and an enol form instead is what functional group? An alcohol, okay? So those are the two forms a nitrogenous base can have. Which form is overwhelmingly preferred? The keto form, good. DNA and RNA absorb what type of light? UV light. And what chemical structure, organic chemistry explains this observation? Aromatic, good. All right, I've already forgotten the number, so I will probably have to flip back. But this statement up here, if it's 2.0, what does that represent? RNA, is that right? Good, because I don't get tested on this anymore. The machine tells me. All right, yes, 2.0 represents RNA, 1.8 instead represents DNA. So far, so good. Pretty easy stuff, a lot of memorization, it's only like a few structures. You won't have to memorize nearly as many structures as you did for, exam 1. Exam 1, you needed 20 amino acids. And exam 2, you needed like a bunch of sugars and a bunch of lipids. For exam 3 onwards, the only structures you need are the nitrogenous bases, and you're good. So we shall move along. All right. Any questions on that stuff so far? Okay. All right. Next is the next level of complexity. After talking about nitrogenous bases, we're going to now attach something to it, and we are going to attach a sugar to it. Both DNA and RNA are made of ribose, okay? So we're increasing the complexity here. We talked about the nitrogenous bases, but now we're going to attach a sugar to it, and that is ribose. Specifically, it is D-ribose, and that ribose has a slight modification to it in the form of DNA versus RNA. So let me find a nice picture of it. Don't worry about linear forms. Same thing with exam 2, don't worry about linear forms. This thing on the left here represents regular old ribose. You know about it from exam 2. There are 5 carbons in it. It prefers a 5-membered ring. And both OHs here are pointing down. That's sometimes how people memorize what ribose looks like. Deoxyribose, which is going to be found in DNA, has just one small difference in it, and that is at carbon number 2, instead of a hydroxyl group, it is deoxygenated. That's probably not the exact word, but that's how I memorize it. Is that this is one less oxygen, it has become a hydrogen, okay? That small difference, believe it or not, changes a lot of different things in terms of stability of RNA versus DNA. You might remember in like organic chemistry and maybe general chemistry that alcohol groups, because they're very polar, they like to interact with everything, right? We know hydrogen bonds in general chemistry. We know, additions, elimination, substitution. I don't remember all those different reactions in Orgo 1 and Orgo 2, but alcohol groups like to interact all the time. So RNA, because it's made of regular old ribose, has this extra alcohol group, thus making it less stable because that alcohol group wants to interact with a bunch of stuff. Deoxyribose, which is what DNA is made of, because it's lacking this alcohol group, is significantly more stable compared to RNA. So DNA is more stable simply because of this this alcohol group is now a hydrogen. Okay. So these are the sugar parts. Let's see what it looks like when you put the sugar together with a nitrogenous base. Okay. Let's see. Yeah, let's do this one instead. Okay. So here are all our nitrogenous bases. Well, most of them. They didn't draw in thymine for whatever reason. But here are nitrogenous bases, and here are the, I guess because they're only showing us RNA bases. And here are the sugars that they're attached to. Notice the nitrogenous bases prefer to go here. What position was that? Let's take a look back at the picture of these riboses. Let's see. What position, what phrase did we use in exam 2 to describe this carbon? Anomeric carbon, right? It is the only carbon bonded to 2 oxygens, right? Remember a long time ago I gave you a bit of like a footnote to that statement saying technically the answer for anomeric carbon is something bonded to 2 heteroatoms, right? For exam 2, that only meant 2 oxygens. But we're going to make it a little bit more specific now. What 2 heteroatoms can mean is oxygen and nitrogen. Nitrogen also counts as a heteroatom. So this is still an anomeric carbon, and it is still, it is still defined as an anomeric carbon. And that is, again, because the technical definition is that a carbon bonded to two heteroatoms is called an anomeric carbon, and nitrogen counts as one. So these are all have their anomeric carbons with all their nitrogenous bases attached to them. What carbon number is this? Is it labeled on here? It's not labeled on here. What carbon number is the nitrogenous base attached to? Carbon 1, right? We learned that very often, for a lot of monosaccharides, your anomeric carbon is carbon 1. It's carbon 1 for like most of them, right? Fructose, if anyone remembers, what carbon number is the anomeric carbon in fructose? It's carbon 2, right? We won't run into fructose anymore, but that's a bit of a review for exam 2. Okay. So, attaching a nitrogenous base to a sugar actually changes the name that you would use to describe this. So let's go back to this page to figure out how to name these things. When you attach a nitrogenous base to a sugar, it becomes a nucleoside with an S. Not nucleotide, that is technically something else, and we'll talk about nucleotides later. But if you see the phrase nucleoside, that is nitrogenous base attached to a sugar, okay? And it's attached via glycosidic bonds at the anomeric carbon. If you attach any of the pyrimidines to an anomeric carbon, it changes the name to where the suffix now is idine. So cytosine becomes cytidine. So if you see the phrase cytidine written anywhere on the exam, you now know that that means a cytosine attached to a sugar, okay? Thymidine, so notice the suffix changed to idine, or thymine changed into thymidine. The suffix indicates that this means thymine attached to a sugar, specifically ribose, of course. And uridine is very obviously uracil attached to a ribose sugar. The rules are different for purines. The suffix changes to something else if we're talking about purines attached to sugars. The suffix is now osine, so guanine becomes guanosine, and adenine becomes adenosine. Okay, so in terms of naming, if you see it written like any of these up here, understand that they're talking about nucleosides. Okay, not just the nitrogenous base, they're talking about nucleosides. You see this phrase here, confirmation could be syn or anti. Technically true, I'll explain what that means here. Confirmation could be syn or anti, but overwhelmingly the most preferred form is syn, S-Y-N. And why that is because this huge aromatic group here is actually balanced by that CH2OH. I don't know the really complicated biochemistry 3 reason. All I remember and all you have to know is that the syn conformation is overwhelmingly preferred, where this group here is pointing in the same direction as the CH2OH over there. Okay. All right, so we'll stop for now. We'll continue next time on Thursday. And again, everything is recorded, and I'll post it on YouTube later.